So let me begin by thanking Professor Somdatta and Professor Narendra for this kind invitation. It's a pleasure to be here. It's okay. So I thought I will talk a little bit about how we are using AI, especially the generative AI technologies, for asking some basic science questions. We do have
it's now 20. So
20.
Use the whole thing.
Thank you. Thanks, that helps. Yeah. So of course, AI has been extremely useful in the end stage application, as you heard from brilliant talks from the previous two speakers. We do have in the lab precision medicine based applications, but I thought today I will tell you one story about how we can use a basic question using AI. Much of the work that I'm going to talk about is done by my graduate student Arnav from the bioengineering stream. So this is, I'm going to tell you about how we decode immune response patterns, the complexity that are there across diseases, using, of course, physics-based information, large data, especially transcriptomics, and AI models. Right? So let me begin by just reminding all of us, I mean, this is not new to anyone, I'm sure. So what the fact that humans are afflicted with a variety of diseases. And when you think of disease, the first thing you think of is the organ, perhaps, the symptoms, which doctor to go to, because it's so very different, and then, with age, perhaps the cost and practical aspects like that. Let me give you a different perspective. The whole lot of diseases, the point was not to give you a lot of information on this, it's just to remind ourselves about the space that we are working with, really. So thousands of diseases, out of that maybe hundreds are well characterized, and some of them are very frequent. Some of them don't even have a name, and you just assign some name to it, depending on various other things and so on. I want to give you a different perspective, a molecular systems perspective of disease. Something like, perhaps more appealing to engineers, I would think, than conventional biologists, but it is nevertheless very important to think about it like this. And now we have the means to think about it. It was not possible, let's say when I began my career, people would have laughed at me if I had started with a question like this, because it was just not practical to do that. But today we can actually do this. Right? You think of it as a box, not a black box, as a transparent box perhaps, where something senses the pathogenic triggers, right? Whether it's a mutation that's happening in cancer cells, or it's a pathogen like a virus or a bacterium or whatever that may be, and molecules from that. So that's a trigger, and then something senses that and detects the trigger and relays that information. All of these are to various protein molecules that are there in our human cells. So to responder machinery out there who then respond. I mean, this is oversimplification of the whole process, but that's what it is. And what happens at this molecular level must be felt at the cellular level, at the tissue or organ level, which is what actually gives rise to symptoms at the organismal level. So the clinical manifestation that you see is a result of all of these things that are going on. Whether it's in a model system like a mouse or in a human, it's the same story. So I would like to think of a disease as a function of molecular aberrations at the tissue or organ or whatever, and physiological perturbations that goes on, which are symptoms. But the story is very complex because there is a pathogenic trigger. It's not a linear pathway that goes into aberrations and symptoms, because even when there is disease, people are still alive, cells are still functioning, metabolism is happening, you're thinking, eating, drinking, whatever, all that is happening. So there's compensation that is going on, the local perturbation, the global perturbations. There's a compensation, homeostatic correction, repair, and so on, and metabolic rewiring, reprogramming, and so on. And the outcome is a combination of whether there's perturbation, compensation kind of a thing, and recovery, and whether it goes into a chronic state that we live with. I don't know how many chronic illnesses each of us have. We really don't know. That doesn't manifest actually as clinical symptoms, and whether it progresses into something active. So why I'm giving you this picture is just to drive home the message of complexity that's out there. And let's just go back one step. We have like 37 trillion cells, something in that order in the human body, and each cell has 20,000 genes. Even if you take only coding genes, orders of magnitude higher number of transcripts of each of these genes, and the number of protein molecules, a large number, many zeros out there. And if you look at interactions among them, it's orders of magnitude higher than that. So it's a very, very complex picture. And this is changing dynamically. We have seen changes within a few minutes after a trigger and so on. So those kind of changes happen. A few minutes or even earlier, maybe. Why the complexity, the homeostasis includes all of these things: feedback, regulation, cooperativity. I mean, I don't mean this as a classroom lecture, but all of these things necessarily are going on out there. And the result of this is emergent behavior, robustness, coherence, adaptability, and so on. Okay? So again, the point I'm trying to make is it is not sufficient to look at single molecules or single pathways at a time. We really need that helicopter image and be able to dive deeper as necessary. So systems biology is really essential. And the way to do, one of the ways by which you can do systems biology is collect large data, which fortunately is now possible with all the omics and related technologies, make mathematical models, simulate it, and then do that. So this was what we have been doing in the last 20 years, in fact. In fact, it's, people talk about fourth paradigm or fifth paradigm. It doesn't matter what we call it. So we want to harness the power of big data, bioinformatics, systems modeling, and AI, all in sync. Okay? So at the end of the day, I'd like you to just remember that they're complex patterns, and recognizing which pattern is associated with what, or identifying these patterns, is one of the goals in this field. So with this, equipped with this kind of understanding, now we thought we will ask very specific questions about diseases. The same human system responds to a variety of pathological triggers. We are able to come up with a response to different types of diseases and then solve it to various levels, right? The same set of 15,000 to 20,000 genes present in the cells respond to these triggers, and each person who sees this trigger responds to it in a very similar way, which is why we can recognize the disease as a disease. Of course, there's heterogeneity within that, and there are subgroups, and that's where precision medicine comes in. But nevertheless, you can recognize diabetes as diabetes and cancer as cancer. So what gives rise to such precise molecular perturbations in the cell? And how do our cells sense these pathological triggers and make sure that the downstream processes are so precise? How do they transmit? What is the mechanistic understanding of this? How is a phenotype produced very precisely? That's what we're trying to show in the image out there. Now we have a problem. We want to study many diseases. Each one is a different organ, different tissue. We can't compare apples with oranges, even if they are human cells. You just cannot compare liver tissue with muscle tissue and then come up with an answer that is scientific. So to compare all diseases, fortunately we have blood as a tissue. Blood carries disease signatures, which you can read out from the transcriptomic data. So that was what, and there are many publications, many papers, a lot of experience in this thing. So if you see the image on the right hand side out here, each column out there is a transcriptomic data of one patient. So the colors actually represent out here whether it's overexpressed, underexpressed, or no change as compared to healthy people. Some of these are healthy individuals. Each column is a patient. Some of these are, I've taken TB as an example because we work a lot with that. Tuberculosis patients out here, blood samples from tuberculosis patients, and these are treated at various time points without telling the algorithm anything about the condition of the person. If you agnostically cluster, you'll see all healthy individuals going into one side and all the TB another side, and the treated ones, depending on the success of the treatment, group somewhere closer to one of the two sides, which means that the transcriptomics contains enough information to indicate the disease status. And this is not just in TB, it happens in cancer, it happens in autoimmune diseases, it happens in metabolic diseases, and so on and so forth. So blood is an accessible and unifying tissue that reflects disease states. Blood transcriptomics successfully classify healthy from disease, and transcriptomes are beautiful. I like them a lot. The measurements of entire genomes. So it enables biology at scale. Now we have many advanced technologies, spatial transcriptomics, single cell transcriptomics and so on, but what I'm showing you is just the bulk. Now let me quickly go into the diseases that we've been looking at. So we obtain blood transcriptomics that are publicly available, because we have small data sets here and there that we got from clinical studies, but that's nothing in size as compared to what you have out there in the databases, so that's what we take. So about 53 different diseases out here, many data sets, total number of samples in thousands, including controls, pathology matched controls, healthy controls and so on. And these data set represent several major disease types, so we thought it was a good starting point to do that. The first image that you see is based on what's called disease ontology. It's another database where people look at the organ, the etiology of that and so on, and then come up with anatomy-based hierarchical disease classification. So we asked a simple question: if we take transcriptomes, will we be able to reproduce this classification that's well accepted in the medical world? Okay? And indeed, it works out quite well. Of course, we have different measures for comparison and so on, which I will skip in the interest of time. So that was the starting point. Now, we wanted to leverage, to ask these questions, which was so complex, we wanted to leverage the system. I mean, we were able to do that because we had systems modeling methods in the laboratory at different levels in the biological hierarchy. I won't dwell on them, but for each purpose, for each sub-question, we have a modeling type that is available. Many of them are available publicly, and a large number of people who have worked on these in the world. So the method that we used quite heavily here is to use a knowledge graph of protein-protein interactions in the cell, which includes physics-based interactions, includes bioinformatics predicted over the physics information. So it's somewhat like a network, a large network, a roadmap of a large city like Bangalore, a messy city, I would say, in terms of traffic. Something like that is what you see. And to mine out very specific interactions that are relevant, is a challenge. So we came up with graph theoretical algorithms which will tell you what are the changes in this big city, let us say between 8 a.m. and then 6 a.m. in the same city. Where is the traffic going? Where are the traffic perturbations that are out there? So our algorithms capture some of those and then get insightful ideas about which pathways and so on and so forth. So what we had as a number of publications from the lab on this. So what we had as a starting point was RNA-seq data for each of the diseases, contextualized graphs, genome-wide knowledge graphs, and this is the perturbation networks that came out of the algorithm. And even this was so complex that we could not make sense out of it by manual inspection and so on. We also have functional enrichment and so on. So what we found from this was, across all diseases, immune processes ranked as top perturbed, and out of that it was the innate immune pathways that were top perturbed. So if you see these things out here, they're present in more than 90% of the diseases, even if they are not infections, right? So that is what you actually see. So a human innate immune system is well understood. There are a number of pathways, it does a lot of functions, but one thing that I want to draw your attention to is the textbook image says it's non-specific protection that detects danger and so on. So is it really non-specific is a question that we asked, and then we wanted to understand if innate immune system is at the common core of all of these diseases, can we identify if there are any central players in them, right? Who is allocating basically a response pathway to this particular pathway or that different one and so on and so forth. Are they central players is a question we asked. To address that, we came up with this AI method, a graph neural networks. In brief, a graph here means the protein interactions for us, and the weights of that initially informed by transcriptomics data, but we want to learn based on attention, just like I think attention is a well understood problem now. Can you go on increasing the weights if they are important for the problem to solve? So that's what we are actually looking at. So it's a big improvement over traditional GNNs, the attention-based ones. And we thought we will do it just like humans learn, do it at different levels, go to primary school and then to secondary and then graduation and so on, and that's what exactly is happening out here. There are different levels of this is going on. Okay. So Arnab actually devised a GNN, a graph neural network at different layers, 3 different layers out here, I will keep the technical part short. And at the end of the day, we were able to identify a short panel, a small panel of 23 genes as central tuning factors. Now, these tuning factors are not direct sensors like pattern recognition receptors and so on, but they are central. If you change their expression levels, you get a different tune pattern. But it's a combination of them. It's not a single one at a time. Not all 23 are present in everything. A subset will be present in any given disease. Now, we asked a question, if we just take this 23, can we reproduce the classification pattern of diseases? And that's exactly what you see here. We call this T23 set. These 23 are central, and they are the most influential, which is what was identified by the AI algorithms. They reproduce largely the same clustering pattern that you had with the disease ontologies or with the whole transcriptomes, indicating that, of course, there will be some differences. It's not identical. It may even be better in some ways, but it is sufficient to get this broad classification going. So that was really interesting for us. Now you ask, what are these 23? How do you actually make sense out of it, and so on? So we looked at a topology-based node annotation. Where are they present in this network? Are they in the central core? Are they in the next neighborhood, primary interactors, or are they satellites somewhere along the way? First satellite, second satellite, and so on. So this was those are the colors that you see out here. Examples of them, I've taken again tuberculosis and cystic fibrosis, which are very different diseases really, but there are some central players in red. These are gene names, so that are common to both of them. The same genes are present, but their neighborhoods are different, which is why there are such different diseases out there. In fact, the tune patterns provide insights into the differential pathogenesis mechanism out here. It's just a little snapshot. These are different genes and so on. It may not make any big sense to you from far, but they're very insightful because each one has a gene name, an interaction, and which is these red ones out you see out here are the nodes which are part of this 23 gene panel and so on. So in fact, TB shows a system where compensation can genuinely win because it can push it into latency, whereas cystic fibrosis shows a system where the initiating lesion structurally prevents compensation, because even the neutrophils, antibodies, the adaptive immune responses can become pathogenic. So we are able to dissect pathogenic from protective consequences that are going on in this system. So that can form a separate question, which we plan to take it up in the future. But you get like a very, very insightful, detailed resolution of information out here to say what is the trigger, who are the responders, and what is the response, who are they relaying it to, and what is the action that you see at the tissue level and at the organ level. So in fact, let me stop with a summary here. So what our study has of learning from pan-disease transcriptome, what it shows is that innate immunity has specificity. It's a common thread across major diseases, and it's well supported, of course, by converging evidence. You'll have to synthesize the evidence from individual studies because nobody is asked this question. It is, like I said, textbook information that innate immune responses are boring. They're important but boring. There's nothing exciting about them. But that's not really the case. In fact, in a separate instance, we have T-cell mediated, CD8 mediated immunity based work in the lab for cancers. And we are seeing differences in survival when innate immune response triggers CD8 responses in a certain way. Or whether they push them to exhaustion or whether they push them to being active and so on, and that has a survival implication out there. So for this, again, of course, innate immunity and inflammation go hand in hand, and if you can think of innate immunity as the sensors, sense and respond, inflammation is like one of its most visible outputs. Okay? Now, there are GWAS, genome-wide association studies, where you associate genomic changes to transcriptomic changes available for many diseases. And again, we find that immune inflammatory pathways are enriched in almost all diseases, so which ties up with the data that we are looking at. So our study does indicate specificity in innate immune patterns, not individual genes, but as a pattern, as a subnetwork, with each disease exhibiting a characteristic pattern. And we find 23 to be at the core, and they can be called as tuning factors, which gives rise to specific tune patterns. And then as a last comment, I want to say that AI technologies have high potential in driving new basic science discoveries and finding generalizations in biology, perhaps as much as applications, downstream applications. Thank you for your attention. Got it. It depends on what you mean by correlation. Correlation is very context specific, right? So our idea was to start unbiased, don't have any favorites, don't assume one hypothesis and chase it, but instead derive hypothesis and later verify that. So that was our idea. So you can do that, but you will require some data for those things. Yeah. Yes.
So very nice talk, Suma. You just have one quick question, which is that you discovered these 23. They are all part of the innate immune system. I'm thinking about application, and you gave an example of cystic fibrosis.
Right.
Why would I apply the 23 transcriptomic signature as opposed to delta F508? That's my question. So when would I use your system?
Right.
You're learning, of course.
Yeah.
So if you have an unknown disease, maybe you can.
Yeah.
But the examples that you gave, we know what the players are. So
Right.
why would a clinician want to do 23?
This was only as comparison to ground truth, right?
So
you have many
what I would
new
have liked to see
findings.
is something that's not known in medicine.
Absolutely.
And you have discovered it using your 23.
Absolutely.
That would be a wonderful
Absolutely.
thing to know.
First of all, you identify the entire process, like the sensors, which combination of genes are basically involved in sensing the pathogenic trigger. And we are also working on quantifying the response of sensing and transmitting and relaying and so on. So these, at this point of time, I don't want to claim that this is applicable directly. This is more fundamental understanding. But what will come out of it, I know because we are working from the other end as well, is applications for subtyping a disease. So looking at what is protective versus what is pathogenic as a consequence. So what is a compensatory mechanism? So, and treatment can be tailored based on that understanding. Yeah.
Thanks for a very interesting talk. How do we validate this? Do you have a plan
Yes.
to, how do we do that?
So it's not a simple validation like we would do for say a single gene biomarker, because this is a systems profile. The validation that I showed you to some extent was how genome-wide association studies have found mutations. Can we synthesize the evidence that is coming from multiple types of data and then put them together? Is there coherence, is what we are trying to ask. And then of course there can be more focused experimental studies to validate these things. But when I sort of come up with an idea of how to do it. Yes.
Good morning, ma'am. I have a question. Like you mentioned, like every disease has its tune pattern, right? So if let's say somebody has two diseases at the same time, so how does the AI model help in differentiating between the two diseases, and how does it like not mix them up?
Right, right. So these are, so that becomes a new type, a new phenotype, which is a combination of disease A and disease B. So again, data is important for any machine learning model to happen. If there is data of, let's say, hypertension and diabetes are so common together, so one has to train with a new label out there, right? And then there are deconvolution methods to do that. It all depends on how much training data you have.
Let's thank Dr. Suma again for a wonderful talk.
Thank you.
Thank you very much.